Wednesday webinar series. Today's webinar is on interindividual variability in transgene mRNA and protein production following adeno-associated virus gene therapy for hemophilia A. My name is also Mirtha, and I am the Manager of Strategic Communications here at the National Hemophilia Foundation. At any point during this webinar, if you would like to ask a question, please use the Q&A feature at the bottom of your screen. If you're joining us from one of our live streaming social media platforms, you can always ask your questions in the comments. We have NHF staff members monitoring these questions, which we will post to our speaker after the presentation. This webinar is being recorded and will be available to the community beginning Friday, January 27th, our last webinar of January already, my gosh. I am joined today by Dr. Sylvia Fong. Thank you for taking the time to join us today. I will now turn it over to you to get us started.
Thank you, Alsan, and thank you, National Hemophilia Foundation, for the invitation to speak at this seminar. I'm very excited to share the findings that we've had over analysis of liver biopsies from participants who received AAV gene therapy for treatment of hemophilia A. And I'm an employee of BioMarin, and here's my disclosure. And also, I will not discuss the hemophilia gene therapy efficacy in this presentation. So, galactose roxaparvovec, or AAV5H human factor VIII SQ vector, is an AAV5 serotype capsid carrying a single-stranded DNA encoding for a B-domain deleted human factor VIII transgene, and that is under the control of a hepatocyte hybrid hepatocyte promoter. As some of you might know, that this gene therapy has been has been used to treat severe hemophilia A participants in clinical trials. And so far, we have 5 years of expression data, as you can see on the right. And the expression pattern is characterized by a ramping up period within the first half of the year after treatment, plateau for some time, and then decline year after year. There is unknowns related to the therapy in terms of the mechanisms mediating the interindividual variability, and as well as what I mentioned earlier, that there is a decline of activity. So the mechanisms that mediating this decline is also of very strong interest to us. So because of this is a fairly novel treatment, we wanted to understand what is happening at the tissue level of the gene therapy participants. And so we conducted a liver biopsy sub-study we offered this sub-study to all the subjects in the phase 1/2 trial of galactose roxaparvovec. And optimally, we would like to include subjects that were dosed at different dose levels, and subjects who have produced different factor VIII activity, including a subject that has low factor VIII activity, so that we can understand the potential mechanisms that's mediating the intersubject variability. We also would like to examine the AAV vector genome forms that's present in the tissue, as we would like to understand how it relates to durability over time. And of course, it would be remiss if we don't look at the safety aspect of AAV gene therapy using the liver tissues, so that we can really assess at the tissue level if there is any safety findings, with the caveat that there is no baseline pre-gene therapy sample for comparison. And we were indeed very lucky that we were able to enroll 5 participants in the sub-study. And so the participants' numbers are on the left of the table. And indeed, we were also able to enroll participants across 3 different dose levels of AAV5 factor VIII SQ gene therapy. And these include 6E12 vector genome per kilogram and 2 participants from 4E13 vector genome per kilogram and 2 from the 6E13 vector genome per kilogram cohort. The biopsies were collected roughly about 2.7 to 0.8 years to 4.1 years after gene therapy. And based on the cores that we've obtained, for each one of these cores, we actually were able to analyze quite a decent numbers of hepatic globules within each of the biopsies. At the time of the liver biopsy, all of the ALT levels were normal. And in terms of the factor VIII levels at the time of biopsy, as I show on the right-hand side of the table, let me take you through this one at a time. So participant 1, he was dosed with 6E12 vector genome per kilogram, and that's the lowest dose that we've treated our subjects with. And he's never had an appreciable factor VIII activity after treatment. So his factor VIII activity at the time of biopsy is the low limit of detection. Participant 11 and 15 were both treated at 4E13 vector genome per kilogram. And participant 11, as you can see, that he, at the time of, before the liver biopsy, he has about 18.6% of factor VIII activity as measured by chromogenic assay. And if you were to measure it by one-stage assay, his factor VIII activity was 28.4%. But in contrast, in participant 15, at the time of liver biopsy, he didn't really have much of the factor VIII activity as measured by both assays. And in fact, this participant has really never produced much meaningful factor VIII levels after gene therapy. Now, the last 2 participants here, participant 3 and 4, and this is 4 years, more than 4 years after gene therapy, and you can see that they both still have roughly about 10 to 20% of factor VIII activity, depending on the assay used. So the first thing we did is we looked at how the AAV gene vector transduced human liver. And as you can see here, with the images shown on the left-hand side, these images are in situ FFPE tissues that have been subjected to in situ hybridization, specifically detecting the vector genome from galactose powder bag. And these brown dots in the nuclei represent the vector genome that's present in the hepatocytes. So you can appreciate that there is quite a bit of those brown foci within the nuclei throughout the hepatic globules. And if I were to transcribe the findings to a more quantitative measurements as shown on the right-hand side, by showing percent positive nuclei that contain the vector genomes, and you can see that there is a dose dependency of increased number of hepatocytes that are staying positive for the vector DNA. And for 4E13, those participants, we have about 32% of the hepatocytes that contain the vector DNA. And for the 6E13 dosed participants, about 50% of the hepatocytes have staying positive for the vector DNA. But with this staining, we don't really know if the DNA that's in the hepatocytes are functional or they are full-length genomes, or how they are present in the liver, in the hepatocytes. So with that, what we did next is employing a molecular technique called droplet digital PCR to quantify the number of full-length circular genome. And the reason why we're interested in it is that, as I mentioned earlier, AAV gene therapy, these capsids contain single-stranded DNA. And after IV infusion, it's taken up by the liver target tissue hepatocytes. It will go through a series of process of being trafficking through the intracellular space and then to into the nuclei. The DNA will be encoded inside the nuclei, and the single-stranded DNA will have to find the other partner of the either the negative strand or the positive strand of the DNA and assemble into full-length DNA in order to provide transcriptional activities to produce RNA of factor A, and then a protein being folded and secreted and to the circulation. And so that's why we're interested to understand what is the type of genome that's present in the nuclei. As I show you in the previous slide, that there's quite a bit of number of hepatocytes still contain the DNA in the nuclei. And so by utilizing a droplet digital PCR, we're able to quantify the full-length genomes that's present in the hepatocytes. And these genomes, when they are in the nuclei, over time, they become fused into the circular structure called episomes. And so they have these inverted terminal repeats, and that's where the episomes are fused. So we also assay whether the episomes with ITR fusion also exist in the hepatocytes. So by doing this molecular analysis, what we can see here on the upper left-hand side of the slide is that we indeed see it again, a dose dependency in the number of full-length genomes that's present in hepatocytes, and they roughly correspond to the number that are ITR fused, and that means that they're circularized and remain in the nuclei as the stable episomes, as how AAV vector persists in a number of years. And then we also employed a alternative orthogonal method to visualize the genomes based on the size and the form and structure of those genomes by using a southern blot analysis, as shown on the lower side of the slide. So when we run the DNA from the participants' liver biopsies and analyze the form of DNA that's present and corresponding to the size of the genomes, and we can see that in the participants that we assayed, except for participant 1 who was dosed with a very low dose, all the participants from the 2 other cohorts all have the episomes present as a monomeric form as well as a dimeric form. And that there are also higher molecular weight form that you can't really see very well on this image, but they also are there. But what I would like to draw your attention to is participant 15. As I mentioned earlier, he never really mounted much of the factor A expression following gene therapy. But in the previous slide, I show you that he also has similar number of hepatocytes transduced as participant 11. And in the case of molecular analysis, we also see similar number of full-length genomes present in his liver biopsy. And the number of genomes that are present are pretty reasonable and decent. For the 4E13 vector genome per kilogram dose participants, we have roughly about 1.5 copies per diploid genome, whereas for the 6E13 vector genome per kilogram dose participants, we have about 3 to 4 copies of the full-length genome. And these full-length genomes are presumably capable of giving rise to transcription. Now, the next thing we did was to look at then the next step. Since we can detect genomes that are present in the liver biopsies, what about RNA? So we next analyzed the amount of RNA that's present in the liver biopsies, and on the upper side of the slide, you can appreciate that again there is a general dose dependency of the amount of mRNA that's present in the liver. However, in participant 15, the low responder, we can see that the factor A SQ RNA that's present in this participant's liver biopsy is fairly low compared to the responder participant 11. And we then did additional imaging analysis by using, again, in situ hybridization to detect the factor A SQ mRNA in the cytoplasm. And that's what's shown here with these purple dots. That's shown by the arrow here. And we were able to quantify the amount of factor A mRNA that's present in the hepatocytes. So what we show here is that in the non-responder participant 15, compared to the responder participant 11, in his hepatocytes, there's both less number of hepatocytes being positive for the mRNA, and for the hepatocytes that were producing mRNA, there's also less of mRNA that's being produced. And overall, the molecular analysis is measured by ddPCR in terms of the amount of RNA produced correlates very well with the orthogonal method that we use to quantify the amount of signal within the liver biopsies using in situ hybridization. So from this analysis, we conclude that for the non-responder or the low responder in this cohort of participants, the reason why he is not producing much of the factor A activity is because he didn't have much of the factor A mRNA in order for the protein to be translated. And we then conducted additional molecular analysis using RNA-seq with the remaining RNA samples that we still have. And in comparison of the differential expression pathways between the responder, participant 3, 4, and 11, versus the non-responder, participant 15, we noticed that the molecules that are involved in the positive regulation of transcriptional pathways are downregulated in the participant 15's biopsy. And in particular, we noticed a particular gene called PHF5A. The expression of PHF5A is much lower in participant 15 comparing to the other participants. And intriguingly, PHF5A is involved in the transcriptional elongation by RNA polymerase II and pre-mRNA splicing. And this indeed implicates a role potentially for PHF5A that could play in the participant 15's response in terms of low mRNA production. And this molecule was not a stranger to us. In fact, in our preclinical studies, when we were looking at correlation between expression of factor A RNA in the gene therapy treated mice and correlating that with PHF5A expression, we can see a significant correlation between the expression of PHF5A with the amount of factor A mRNA production in the mouse liver. And if we do additional studies to in vitro studies to understand the role of PHF5A in factor A RNA production from the AAV gene therapy vectors, we use a knockdown technology to knock down the expression of PHF5A and in a human hepatocyte cell line. And what we show here on the right-hand side is when we knock down a PHF5A expression, we then knock down the ability of the cells to synthesize mRNA from the AAV vector genome. Now, factor VIII protein, as some of you might know, is known to be an inefficiently folded and secreted protein from the endoplasmic reticulum. And the gene therapy construct that we employ here is a B domain deleted factor VIII transgene. And so it translates to a B domain deleted factor VIII protein. And similarly to the full-length protein, it is also inefficiently folded and secreted from the ER. And previous preclinical data have suggested that in vivo hydrodynamic delivery of plasmid that encodes either full-length or B-domain deleted factor VIII proteins could induce unfolded protein response when there is a lot of input of DNA at a short amount of time. And more recent data also in mice show that there is some evidence of unfolded protein response or ER stress response in animals treated with the B-domain deleted factor VIII transgene with a factor VIII vector, which is much tropic to liver mouse hepatocytes, or in the case with a very strong promoter. Although in those cases, there is not indications of hepatotoxicity. So what we're interested in learning from the liver biopsy is, are there any ER stress that can be detected in the cells that are expressing the B-domain deleted factor VIII from our gene therapy? So the hepatocytes, as you've shown here on the left-hand side, is an image, is a confocal image that we took of representative cells that are expressing factor VIII protein that's represented by the red dots. And we can then detect whether or not there is any elevation of a ER stress marker using GRP78 as the ER stress marker, and within the cells that are expressing factor VIII. So using the quantitative analysis after a confocal microscopy, we can quantify the amount of ER stress marker signal, GRP78, in the cells that are expressing factor VIII protein versus in the cells that are not expressing factor VIII protein. And the results are quantified on the right-hand side of the histogram. And as you can see here, indeed, that there is no difference in terms of the ER stress markers between the cells that are expressing factor VIII protein versus the cells that are not expressed factor VIII protein, suggesting that there is no ER stress at the time of the liver biopsy. And then we, of course, as I mentioned earlier, we were interested in understanding if there's any safety signals following AAV gene therapy, and particularly long-term safety concerns. And so we did a histopathology analysis, and the histopathology analysis is done as the following manner. The local sites will first do the histopathology analysis with the local pathologist, and then all of the slides are then sent to a central pathologist, and the central pathologist will then analyze these tissues, and the findings between the local pathologist and the central pathologist are pretty consistent. Which they found that there is no clinically relevant inflammation or sinusoidal infiltrates in the liver biopsies we collected between 2.6 to 4 years following AAV gene therapy. There is no fibrosis, no evidence of dysplasia or any significant architectural distortion from any of the participants' biopsy analysis. The only thing we observed is our mild steatosis in 4 out of 5 participants, which is characteristic of patients or people who are in Western society. And so with all of the analysis that we were able to do with these very, really precious material, we show that valactocogene roxaparvovec transduced the human hepatocytes dose-dependently, and the genomes are distributed in a panlobular pattern within the hepatocytes. The long-term expression of factor VIII following one gene therapy infusion was associated with the presence of the durable episomal genome between 2 and 4 years following gene therapy. And we did observe inter-individual differences in the transgene expression. And this particular analysis, I show you that there could be potential differences in terms of the ability to express the mRNA despite successful transduction. And in histopathological examination of the liver biopsies following AAV5 human factor VIII SQ gene therapy, we did not see any evidence of endoplastic reticulum stress in hepatocytes that are expressing factor VIII protein, and there's also no evidence of any tissue damage or inflammation that we can observe. And lastly, I just want to express so much gratitude towards the valactocogene roxaparvovec study participants, and particularly for the participants that participated in the biopsy studies, and also the my colleagues at BioMarin that have contributed to the data that I presented here today, as well as the PI that have also administered the gene therapy to the participants that we study in this presentation. And with that, I like to stop and take any questions you might have, any comments as well. Thank you.
That was great. Thank you. Let me just pull up my screen real quick. Let's see. Okay, we have just a quick announcement to make. Just one moment. Sorry, everyone. Okay, recruitment is now open for Own Your Path, a program for young adult males with hemophilia who are currently in the transition years between 18 and 30. We know that maintaining adherence to a prophy regimen can have short and long-term health benefits. This program focuses on helping participants be successful by focusing on education, health coaching, skill building, and relationship building, all within an app-based environment that includes participation incentives. If you have patients in this age range and think it would be a good fit, please email us at ownyourpath@hemophilia.org, and we can send the flyer for you to share. For consumers, you can email us directly and we can provide more information or register you for the program. Okay, so on to the next. I only have a couple questions for you today. So if anyone else has anything that they would like to ask, now is the time to go ahead and throw it in the chat for me. We will go ahead and get started though. What is the biggest takeaway you want the audience to walk away with from your talk today?
I think that there is definitely unknowns related to AAV gene therapy for hemophilia A. However, with the availability of these liver biopsies, we were able to start understanding some of the mechanisms that may mediate these unknowns. And so that, because we're really committed to the hemophilia community, and we would like to understand a bit more as to what these AAV gene therapy are doing in the tissue. And so we'll continue to actually enroll participants who is interested in the study for liver biopsies, and so that we can bring more of the understanding for this therapy to all of you.
Great, thank you. Where can I find more information about your study?
Yeah, so this study that I presented was published in Nature Medicine in April of 2022. We can send a link to the publication, and you will be able to get a lot more information from that manuscript.
Great, thank you. Okay, let's see. Is there any difference between participant 11 and participant 15 in immune response to the vector or the length of steroid treatment?
Not that I know of. I don't think so. They all receive steroids. So in the phase 1/2 trial, the participants were treated with sort of prophylactic steroid regimen, meaning that after 3 weeks of AAV gene therapy, prophylactic steroid treatment was initiated. Although that's for the 60-13 vector genome per kilogram dose cohort. So there is really not much of a difference, and all of the participants we treated have all been screened for pre-existing immunity for AAV5. So they were all negative in order to be enrolled in the trials, and they all developed antibody to AAV5 capsid after treatment. And there's no anti-factor VIII inhibitors in any of our participants.
Awesome. Let's see. So for the next one, do you believe doing a similar study post to 10 years gene therapy would be a value for a comparison standpoint?
Yeah, absolutely. In fact, because I mentioned earlier, there is the decline phenomenon that we're seeing. However, what is mediating that decline? It would be very helpful actually for us to have serial biopsies or longer term biopsies, but more numbers of it at each time point. Then we could really try and begin to understand what's causing the decline. So yes, I absolutely think that in 10 years, it would be great to have more biopsies and also to understand could there be any other safety signals that can be detected long term. So yes, absolutely.
Wonderful. Was there anything that you learned during your study that you were relatively surprised with?
Yeah, so before we started the study, I would have thought that maybe the non-responder, the low responders don't respond because they weren't transduced successfully, because that's most of the time the rate limiting steps of AAV gene transfer. Here we're seeing there is actually good transduction happening, but this host-mediated response to the gene therapy vector that there's individual differences in terms of how they respond to the vector, and so that was a surprise to me. But I think that also points us to clues as to how we can maybe potentially help to produce more mRNA if there are approaches to do so, if we understand why that participant is not producing mRNA, given that he's got plenty of genomes.
Great, thank you. How would you suggest community members find studies like this to participate in?
Well, so for these, I believe that these are all sort of sub-studies from our clinical trials. So for now, this will be sort of related to the clinical trials. I don't know if in the future when we have long-term follow-up, whether that could be a sort of a part of a protocol that we could offer. That I don't really know. We need to have a more internal discussion to see the feasibility of that.
Wonderful. And that's it that I have for questions for you. But is there anything else that you would like to share with the audience today or anybody watching this back later?
No, I just, thank you for the really interesting questions and thought-provoking questions. And I appreciate the opportunity to speak here, and thank you for your attention.
Wonderful. Well, I'd like to thank you once again, Dr. Fong, for taking the time to join us this afternoon. We appreciate your time and expertise. I'd also like to thank each one of our viewers for joining us. Please note that this recorded webinar will be available on Friday, January 27th at hemophilia.org under the Events tab with all of our archived webinars. Also available in the Events tab is our upcoming schedule for our weekly Wednesday webinar series. Thank you all for joining us this afternoon and have a wonderful week.
Thank you.
Bye, everyone.